Hello everyone, welcome back to this channel. The topic that we are going to discuss this time is a very interesting topic. So it is Introduction to Genetics with special focus on the Mendelian inheritance or the so-called classical genetics. Many people, when they hear the word genetics, they readily think that it is a boring and difficult subject. Well, it might be true, but I would say that genetics is a very interesting topic because it has a lot of applications. It can be applied in the field of medicine, to find cure to diseases and even to develop vaccine, just like the COVID vaccine. It can also be applied in the field of agriculture to improve crops and livestock, also in the environment to manage it and to rehabilitate degraded environment. It can also be used to solve crimes and to settle parental disputes and many other applications. But the question is, where did it actually start? It began with Gregor Mendel. He was an Austrian monk who experimented with garden peas in the mid-19th century. He noticed that certain characteristics or traits seemed to be passed from one generation to another. He had lots of these experiments, and it was credited to him. That's why he was given the name Father of Genetics. The Father of Genetics. And genetics is the scientific study of heredity. That's what Mendel did. He studied heredity, which is the passing of traits from parents to offspring. Most often, when there is a newly born baby, relatives will argue to who is the lookalike of the baby. So is it daddy, mommy, lolo, or lola? They would say, it is a combination. He got his nose from the father, the eyes from the mother, the skin from lolo, the height from lola, etc., etc. So, Mendel continued what he did. He was most famous for his work with Pisum sativum or the garden pea plants. Between 1856 and 1863, he grew and tested over 28,000 pea plants. Kung ako siguro yan, baka nag-give up na ako, no? 28,000, that means that he was able to do it 1,000 times as well. And in this photo, look, Mendel is contemplating so seriously. He's probably testing his garden pea. So why did Mendel choose to study the garden peas? For one, they are easy to grow. He also noticed that garden peas do have easily identifiable traits. So when we say traits, these are specific characteristics passed from one generation to another. Mendel also chose the garden pea because with peas, he was able to work with large numbers of samples. So what are those characteristics that Mendel studied in the garden pea? So he studied the height, he studied the flower color, he studied the flower position, seed color, seed shape, pod color, and the pod shape. For Mendel's experiments, the first thing that he did was create a pure plant or true breeding plant. When we say true breeding plant, it means that parents repeatedly only produce offspring with the same trait. For example, a true breeding tall plants will only produce offspring which are tall. So for example here, these are all tall plants, giving rise to only tall plants as well. When Mendel was doing his experiments, he was asking himself, so what if? What happens if he would cross two plants which are true breeding for contrasting traits. For example, purple flowers and white flowers. What would be the result? Wrinkled seeds and smooth seeds, tall plants and short plants, etc. And he was contemplating what would be the offspring's characteristics. Mendel chose the garden pea plant, Pisum sativum, as mentioned earlier, for experimental tests of his ideas about inheritance. Under normal circumstances, the garden pea plant is self-fertilizing. The cross-section shows the gamete-forming structures. Sperm-producing pollen grains form in the stamens. Eggs develop, fertilization takes place, and seeds mature inside the carpel. In some of his experiments, Mendel brushed pollen from a plant that bred true for purple flowers onto the floral bud of a plant that bred true for white flowers. To ensure cross-fertilization, the white flower had its stamens snipped off. The cross-fertilized plants produced seeds. Each seed was allowed to grow and develop into a new plant. Mendel analyzed the flower color of each new plant as visible evidence of patterns of inheritance. So, if Mendel crossed the purple flower with a white flower, what do you think would be the color of the offsprings? This now signals us in proceeding to discussing the Mendelian inheritance. Before we can proceed, there are genetics terms we must know. So the first is the dominant allele. A dominant allele is always expressed. It is represented by a capital letter, usually the first letter of the dominant trait. And then here is the recessive allele. This is the one that is expressed in the absence of a dominant allele. Remember that it can only be expressed when it is purely recessive without the presence of the dominant allele. And this is represented by a lowercase letter. The same letter as the dominant trait, just lowercase. So for example, if you use T for tall, you have to use a lowercase t for short. You shouldn't use like T for tall and S for short. No, we don't do that, but we use the same letter, only the uppercase for the dominant and the lowercase for the recessive. Another is the word homozygous. So the word homozygous means that when an organism has two identical alleles, so whether it's both uppercase or both lowercase, that means it's homo. Homo means the same, homozygous, either both dominant or both recessive. Okay, there is no combination, because the combination then is what we call heterozygous. That is when an organism has different alleles. For example, a capital Y and a lowercase y. Another terms that we must know is the genotype. So the genotype refers to the entire set of genes in a cell, an organism, or an individual. We symbolize it 12 letters. Another term that we must know is phenotype. This is the physical appearance of an organism as a result of the interaction of its genotype and the environment. So this is the description of a trait. For example, tall, short, purple, or white. Remember earlier, we say that capital T for tall. So these letters is what we call the genotype, and the tall is the phenotype. So for example, this is a picture of our cat. It has a white color of fur and blue eyes. These characteristics visible to our eyes are what we call the phenotype. So putting together, we have in here a genotype, which is heterozygous because we have the uppercase and the lowercase genotype. So what are these? These are called the alleles. So allele for the dominant tall and the allele for the recessive short. When we put that together, because T is dominant and the capital T is dominant, the small t is recessive, so the effect of the tall allele masks the effect of the short allele, or this T. So that gives us a phenotype which is tall only. When doing genetic process, you better use Punnett square. This is a grid designed by Reginald Punnett to keep track of the alleles. So what you are seeing in here is a Punnett square. So this is a useful tool to do genetic process. For a monohybrid cross, you need a square divided by 4. So you see here, there are 4 squares. It looks like a window pane, and we use the Punnett square to predict the genotypes and the phenotypes of the offsprings. So this is a Punnett square, and it can calculate the probability that offspring with certain combination of alleles will be produced. Crossing one trait at a time. For example, Mendel crossed a pure tall plant with a pure short plant. And it gives us the first filial generation with all tall plants. Now, this one is showing the effect of the dominant tall allele over the recessive short allele. Now, Mendel crossed through breeding plants that differed for a given character. For example, the color of the flower. So, a monohybrid cross is actually a genetic cross involving a single pair of genes. So, one trait, that's why it's called mono. And then parents differ by a single trait. So, when they differ by a single trait, that is why we call it hybrid. Okay. So, these parents, which are actually pure purple and pure white, were bred. And their resulting offspring is what we call the first filial generation, giving rise to only purple flower. So even when you can see here a little p, this will only show phenotypically a purple color, because as you remember, purple is dominant over white. So purple in its presence will mask the effect of the white. But what happens if we self-fertilize this F1? So that means F1 cross to another F1. That will give rise to the second filial generation, where the offsprings are purple, and here comes the white came back. So generally, it gives a 75:25 ratio. That 75% is to 25% ratio of having the purple and the white flower respectively. Now let's go into the Mendelian principles or laws. The first principle is the principle of dominance. We have been hearing this since earlier. This happens when one allele masks another. One allele was dominant over the other. Mendel concluded that some alleles are dominant and others are recessive. So as long as the dominant allele is present, it will only show the phenotype or the characteristic of the dominant allele. The recessive allele will only be seen if the recessive allele is inherited from each parent. In other words, you must have two recessive alleles to see them. Here is an example of Mendel's experiments, just like what I was talking about. He always found the same pattern. He discovered that even though one of the parent plants had white flowers, all of the offsprings had purple flowers. So why is this so? Because, as the example that we were given earlier, it's because there is complete dominance. When purple is completely dominant over white, so when they are together in one location, there will only be purple and there will be no white. White can only be manifested when it is homozygous for the trait. The second law that Mendel proposed is the law of segregation. So it states that two factors, or the alleles, control each specific characteristic, or the gene. These factors are separated during the formation of the gametes, or the sex cells. In other words, the law of segregation applies only to traits that completely control a single gene pair in which one of the two alleles is overriding the other. So therefore, the law of segregation does not apply to incompletely dominant or codominant alleles. So what do we have to remember about the law of segregation? So first, genes control traits. And these genes exist in pairs. We call them the alleles. Parents would have both the alleles, but their sex cells will only have one copy of each allele. It cannot be the pair of the allele, only one. So that, for example, if dad has blue eyes and blue eyes is recessive, and if we symbolize it with 2 lowercase b, then we will have, or we will find the alleles for blue eyes in the body cell, but during meiosis, it will be segregated into a single allele in here, and each of the sperm cell will only carry one allele. Law of segregation can be seen clearly in a monohybrid cross or in monohybrid crosses. Now let's see an example of Mendel's experiments. So here, when two true breeding plants are crossed, all the F1 offspring display the dominant phenotype. In this case, the purple flowers. Remember that purple flower color is dominant over the white. Now let's see the genotype. You will see that the purple has homozygous allele, so homozygous dominant, and the white has homozygous recessive allele. All the F1 offspring carries one copy of each, so they are all heterozygous for purple. Now, if F1 having the heterozygous allele would undergo here the meiosis 1 and meiosis 2, we can see that their gametes only carries, or all their gametes only carry one of the alleles. Okay. So when we put that in Punnett square, we will see that let me see, in here we have the dominant allele, another dominant, so we'll have a homozygous dominant for purple. Okay, next, a recessive and a dominant, so you will have one recessive, one dominant. We call that the heterozygous, the same in this case. But in this case, you will have a homozygous for the recessive trait. Looking at the phenotype, you will see 75% purple and the white reappeared. So a 25% chance of having a homozygous white offspring. Now let's go to the third law of Mendel. So the law of independent assortment. In this law, factors or the alleles for different characteristics or the genes are distributed to gametes independently, meaning The allele for seed texture is not dependent on the allele for plant height. For another example, the allele for flower color is not dependent on the pod shape or pod color. This is better understood in a dihybrid cross. Now let's take a look at this example. This is a dihybrid cross, which is a cross between individuals that breed through for different versions of two traits. In this case, the traits are flower color and height. So here, the cross will be between a purple tall flower, both dominant, and a white and short flower, both recessive for the trait. So what will be the offspring? All F1 offspring of a dihybrid cross are heterozygous for both traits and display the dominant form of each trait. In this case, this means that they are all tall and purple flowered. Let's see what happens if this F1 self-fertilizes. So when the heterozygous for two traits plant self-fertilizes, it produces seeds with 16 different gamete combination as shown in this Punnett square. So we already have here some supplied genotype. Let's fill in the missing ones in the blank squares. So for example, here we have a heterozygous for both traits. In this square, we'll have the same. And in this square, we'll have heterozygous for flower but homozygous for the height. And then here, heterozygous for both traits. This one here is homozygous for flower and heterozygous for the height. And here, I'm just going to explain again later. Let's fill in all of the squares first. And then let's see the ratio that we will have later. Okay, and then let's submit. Okay, so we have the correct genotype in all the squares. So now, if you all see, for example, all with capital A and capital D, that means that they are purple and tall. So this one is purple and tall, purple and tall, purple and tall, purple and tall. Here too, this, and that. So if we count that, that's 1, 2, 3, 4, 5, 6, 7, 8, 9. That's 9 over 16 purple and tall. Okay, if we see, for example, here with a capital A but all small b, then that means this is purple but short. So this one is purple but short, this one is purple but short, and purple but short. That gives us 3 out of 16 purple and short. But we also have the homozygous lowercase a and with a capital B. So that means this one is white but tall. Okay? White but tall. So here, this is white and tall, white and tall, white and tall. That's also 3 over 16 white and tall plant. And we only have 1 over 16 that is white and short. So those are the possible genotypes of the cross between the heterozygous F1 plant. Well, that's all for this video. Thank you very much for watching. I hope you got something from this discussion. And if you have questions, feel free to leave your question in the comment section. We will have a separate discussion for the use of the Punnett square for monohybrid cross, dihybrid cross, and more examples. If you are new to this channel, consider subscribing and hit that notification bell so that you will get updated of our new uploads. Thank you very much and bye-bye.